So basically, as you know, from Faculty of Science, Assam Downtown University, we have been trying to organize various lectures from eminent scientists from across India and even from out of India on either online mode or on physical mode. Going forward with such lecture, we had a discussion at faculty level wherein we had thought, why don't we invite some of industry experts so that they can give you an insight of how different processes are carried out, what is expected into industries going out of your courses, because all these people who comes and delivers lectures, and us, we have also been a student in the universities and colleges, and coming out of there, how to shape your future, and looking forward to your dissertation work that you have to do or that you have been doing, there are a lot of analysis that you need to do. And while doing so, you need to understand the different processes that are required for analyzing different samples that you have carried out in your processes. So keeping all those things in mind, we had a discussion, and we are fortunate that we have been having a cordial relation with representative from Walters India Private Limited. We had a discussion with them, and they had accepted our invitation to come and deliver a talk on the basics and advancement in liquid chromatography technology. Now, I believe we have master students here, and more or less all of you must have had a bioinstrumentation chapter, a bioinstrumentation course wherein you must have studied about chromatography. And more or less it has been theoretical, and the chromatography that you must have done would be thin layer chromatography and maybe 1 or 2 column chromatography. So liquid chromatography is an advanced instrumentation technique that helps in and that helps in identification of different compounds. First of all, separation, and then after we pair it with mass spectroscopy so that different compounds can be identified with the help of a library. So that basically is a gist. And in your various researches that you are doing, many a times you need the help of this type of techniques for identification of different compounds and metabolites that you need to identify in your studies. So in line of that, we had invited 2 speakers from Walters India Private Limited. I am very happy to introduce to you Dr. Vishal K. Katavkar, sir. He is the Marketing Development Manager at Walters India Private Limited. Before I call him onto the dais to initiate or begin his lecture and talk, I would like to introduce him to you. He is an MSc in Analytical Chemistry from University of Pune, and he had completed his PhD in Natural Product Chemistry from Jamia Millia Islamia University, New Delhi. And his topic for his PhD was isolation and characterization of bioactive compounds from Millia azadirachta and its efficacy. Now, hailing from Northeast India, we are blessed with natural resources. Being from the foothills of Himalaya, we have a lot of natural herbs that we have been working on, not only in our science laboratory, but also in the chemistry and pharmaceutical science laboratories. We have been working on different natural products. So that being one of the focal point of our research at Assam Downtown University, we had thought of inviting someone in this field so that their expertise can help us as well as the students who are pursuing studies, because most of the faculty's research are aided through the student dissertations. So Dr. Vishal is associated with the analytical industry for past 20 years, and he has been working in various capacities across diverse areas, including agrochemicals, commercial testing laboratories, and as an application scientist. He has been working with Walters since 2008 and has exposure to the latest analytical equipments like liquid chromatography and mass spectroscopy. He has been working closely with scientists on various analytical challenges on liquid chromatography, mass spectroscopy, and audits and compliance amongst others. Dr. Vishal has been part of many scientific forums, conferences, as speaker on various analytical topics such as advancements in chromatographic techniques, basics of mass spectroscopy, and its applications. He was the Northern Indian marketing manager for 10 years and was responsible for promoting new technologies and providing technical know-hows and supporting scientists in their ever-increasing complex challenges with Walters Total Solutions. In his current role, Dr. Vishal is responsible for market development across India for academic and research agrochemicals, specialties in chemicals, and other associated industries. On behalf of Assam Downtown University, we would like to welcome Dr. Vishal. May I request my fellow colleague to kindly felicitate Dr. Vishal before I call him onto the podium. I'd like to request Honorable Dean to kindly felicitate Dr. Vishal. We are also privileged to have Dr. Devasis with us. I'd like to call upon Dr. Devasis, and I'd like to request our Dean again to kindly felicitate him with a gamusa. I believe Dr. Devasis will also be discussing with you about certain things of LCMS. Now, I'd like to hand over the microphone or podium to Dr. Vishal so that he can start with his presentation. Thank you. And we'd like to request all the students to kindly be attentive and listen to him, and we are open, he is definitely open to questions that you may have in you. Thank you.
Thank you once again. And I would say on behalf of Waters India, I would like to thank the Dean as well as Doctor for his coordination, as well as all of you here today for giving us an opportunity to interact probably with the young and bright minds of India, right? Yes. So in the next 1.5 hours, almost 90 minutes or so, we'll be discussing about interesting technologies in the analytical sciences, we can say. That is about the liquid chromatography, and then another presentation we will be discussing with respect to the mass spectrometry. Okay? Am I audible? Yes? Okay, good. Now, am I audible? Yes? Okay, good. So, now before we begin, when we talk of the or think about the future, okay, going forward, like as we were discussing, like most of the faculty is also there, some of the faculty as well as a lot of master students are there, as well as the PhD students are there from different streams, right? So we will be basically focusing on the application aspects, like how we can make use of the technology going forward and how it can be useful to you for your research applications. Okay? Okay? So when we talk of liquid chromatography analysis, okay, like as sir was mentioning, so you might have interacted with liquid chromatography theoretically. Yes, no? Yes, थोड़ा जोर से. खाना खा लिया सबने? Good? Okay. So थोड़ा जोर से. You have interacted theoretically at least, right? So probably after 3 years, after 5 years, when you're ready to pass out, okay, from this college, either after your master's, after your PhD, so you will be either going in the industry, right? That is one aspect. Second, you will be either going in the research area, either as a faculty or in the research area, right? So wherever you go, analytical technologies probably the field in which all of us are working, right? So it will not leave us, right? It will be with us. So probably we will be discussing about what are the basics, little bit of basics from the industry perspective, what they are expecting from us when we go to the industry. When you go to the industry on first day, after 6 months, the expectation is that how fast you are able to learn the practical aspects, right? Because we interact with a lot of industry experts, the industry, so they always have this challenge and apprehension that the students, freshers who are coming, so we need to take time to train them, right? It takes 6 months, 1 year, like that. So how we can make, as I mentioned, the bright and young minds, right, which all of you are. So how we can make all these events, what we are doing. So the major focus is on how we can make it beneficial to you going forward so that it can be useful to you. Okay? So when we talk of basics, technology. No, no, it is not about slide changer. It is about the laptop not getting moved. Yes. So when we go back 60 years, okay, you might have heard about gel permeation chromatography, GPC. सुना है किसी ने? Yes, no, yes, थोड़ा जोर से so that I can hear. Yes, gel permeation chromatography. So around 1958, 1960, the founder of Waters, Jim Waters, he collaborated with some of the scientists from Dow Chemicals to come out with a polymer applications in gel permeation chromatography, GPC. Okay, and that is the reason probably Waters was the first one to come out with a system for gel permeation or size exclusion chromatography analysis. So when we look at the role of liquid chromatography, the role of HPLC, the role is very, very diverse. Right from the synthetic chemistry applications, routine chemistry, pharmaceutics, natural product chemistry, botanicals, nutraceuticals, which is again a upcoming field, right? Biofuels, biodiesels, and all of us know what this is, right? Yes? Anyone? Thoda louder. Vaccine? Yes? So, everywhere the role of liquid chromatography or liquid chromatography coupled with mass spectrometry will be there. Okay, so the applications are very, very diverse. So we'll be discussing a little bit in detail what are different kinds of LCs which are available, then what are their applications, where it can be useful. And mainly when it comes to applications, we'll be focusing on the mass spectrometry aspect. So when we talk of liquid chromatography, okay, so the pump configurations, this is the basic of LC. There are two kinds of pump, isocratic and gradient, okay. Anyone, what do we mean by isocratic? Anyone, have you heard? Little bit louder so that I can hear you. Anyone? What do we mean by isocratic? What is the meaning of iso? Single? Similar? Okay. What about isolation? Single? Yes. So isocratic basically is single, wherein we are using single solvent system. We are not changing the composition of that solvent system. That is isocratic, as the name suggests. Iso means one. Okay. So isocratic. Then another configuration is gradient. Okay. So what does gradient mean and what is its actual purpose? So when we say gradient, the use of gradient basically, or the definition is that when we change the composition of solvent with respect to time, where time is changing and the composition of solvent, if we are using a mixture of acetonitrile and water, so that composition, initially if we are using 90% water and after 10 minutes we are using 90% ACN or methanol, so that composition is changing with time. That is gradient. Okay, so isocratic means one single solvent or mixture, but we are not changing that composition, whereas gradient is we are changing the composition with respect to time. So what is the purpose and objective of using a gradient system? Okay, these are all basic questions which usually are asked in interviews. Okay, so the purpose is that if we are not using a gradient system in our compound or sample, if it has got multiple components, if it is a mixture, so if we are not using gradient, what will happen is the run times will be very, very large. It will take more time for the elution. So when we are using gradient, basically the elution will be much faster and separation will be more efficient. So that is the major purpose of using a gradient system. Okay. And then in case of gradient, there are two configurations. One is low pressure, another is high pressure. So when we say low pressure, it is a quaternary gradient system. Anyone, what is quaternary? What do we mean by quaternary? Yes, 4. So we can use 4 different solvents at a time. Mainly these kind of systems are used in the pharmaceutical or other associated industries. The reason is that the efficiency has to be there. Whereas if we look at high pressure gradient systems, it will consist of 2 or more pumps. Usually these kind of systems are much more rugged, much more robust to use, and these are mainly used for research applications. So high pressure binary systems, what the difference is, if you analyze your sample on a low pressure system and a high pressure system, it is not that you get 2 different results. You get same results. Okay? ऐसा नहीं है कि आपको अलग अलग रिजल्ट्स आएंगे, हाई प्रेशर अच्छा है या हाई प्रेशर खराब है, लो प्रेशर पे अच्छे रिजल्ट आएंगे. यू गेट सिमिलर रिजल्ट्स. द पर्पज इज डिफरेंट. हियर द मिक्सिंग इज टेकिंग प्लेस आफ्टर द पंप्स. So we are having pumps. Pump will be having high pressure. So when the mixing is taking place after the pumps, it will be at a higher pressure. So these are known as high pressure gradient systems. In case of low pressure quaternary systems, the solvent mixing is taking place before the pumps. Okay. So before the pumps, what is happening? The pressure is low. There is less pressure. So we say these are low pressure. This is high pressure. Okay, clear? Moving on, how the actual system will look like. If we look at isocratic systems, we will have a solvent reservoir, a single pump will be there. Isocratic, we discussed single pump. Okay, so we have to either mix 2 different solvents and use them. We cannot change the solvent composition. We can have a manual injector or a auto sampler. We will have column oven and then our choice of detectors. Okay, so this is isocratic. When we move on to binary systems, okay, this is high pressure binary. Did you spot the difference? Yes. So here we are having 2 pumps. There will be 2 separate solvent reservoirs. Solvent reservoirs, 2 separate pumps. After the pumps, there will be a mixer. Okay, so mixing is taking place after the pumps. Then we have the same configuration, the manual injector or auto sampler. We have a column oven and a choice of detector. Okay, this is high pressure binary system. Binary is 2 solvents. Here we can change the solvent composition with respect to time. We can program the pumps accordingly. This is low pressure quaternary gradient system. Quaternary is 4 different solvents, A, B, C, D. Here we are having pump, and before the pump there is a proportionating valve or a GPV, gradient proportionating valve, which basically is controlled electronically, and it takes the solvent according to what we are programming. So it can mix 4 different solvents at a time. Okay, so this is called as quaternary gradient system, and because the mixing is taking place before the pumps, so this is low pressure, and rest of the configuration remains same, like we have auto sampler or manual injector, column oven, detector, everything will be same. Okay, so the difference is in the pumping mechanisms. Okay, so isocratic, binary, then quaternary gradient systems. Okay, then coming on to some more basics, because these are very, very important when we will discuss applications, and we will discuss mass spectrometry. So how to choose what application and what technology. So it will be based on the behavior of the compound of interest or sample what we are analyzing. In case of liquid chromatography, the polarity will play a major role. Okay. So here we say like, likes, like, which means that polars, they are attracted to other polar compounds. If my compound or sample is polar in nature and my mobile phase is polar in nature, so it will come out fast. If my compound is non-polar but my mobile phase is polar, so what will happen? Compound will not elute out. It will elute very slow. So based on the chemistry of the compound of interest, we choose what kind of mobile phase has to be used. Okay. This is the polarity scale for the mobile phase. In liquid chromatography, mainly we use 3 major mobile phases. The most important is water, okay, or aqueous. Second is acetonitrile, and third one is methanol. Almost 90 to 95% of our work is done with these 3 solvents only. We use different additives, different buffers, inorganic and organic both in aqueous phase to make it, to make our compound elute out much faster. But these are the 3 major mobile phases which are used. And if you look at the polarity of these, water is the most polar compound, yes, and hexane is the most non-polar compound available in liquid chromatography. Acetonitrile, THF, etc., they sit in between. So my mobile phase is clear. So mobile phase, water is the most polar, hexane is the most non-polar compound. Now when I move on and look at my stationary phase, what is my stationary phase? My column. Okay, the columns which we are using in HPLC, these contain silica. Okay, anyone? Silica is polar or non-polar? Anyone? What is silica? No answer? The answer is on the screen. Polar, right? So silica is polar. Okay, what is silica? Anyone? Quickly. Have you heard silica earlier? Yes. What it is? How many of you have gone to any beaches? Beach? Anyone? Yes? Lot of people? So what is there on the beach? Sea? Sand? Yes? So what is silica? Silica is nothing but sand? Yes? So, does sand dissolve in water? Yes, no? थोड़ा जोर से so that I can hear. No? Which means silica is polar in nature, right? Water is polar, silica is polar, right? And now I am using different compounds. I want to separate different compounds also, right? So in that case, I cannot only use silica all the time as a stationary phase. So what I do, I take silica, I put a C18 chain, I treat it chemically, I put a C18 chain on the silica, SiOH jo bond hai, so instead of that H, I put a C18 carbon chain. So what will happen? It makes that silica non-polar in nature. Okay. So if I have a C18 column as a stationary phase, it is non-polar in nature. Okay. Silica is polar, C18 is non-polar. Similarly, if I have a C8 column, it is mid-polar in nature. So my mobile phase is polar, stationary phase has to be what? polar or non-polar? Non-polar? Why? My mobile phase is polar, okay? Water or mixture of water, methanol or water HCl. Now what should be my stationary phase? What should be my column? Should it be polar or should it be non-polar? Non-polar? So what is the purpose? What is the reason why we are taking non-polar column? The purpose is to separate my compound. Okay. My compound will behave, it can be coming out or it will be soluble in a polar basically context, or it can also be soluble in a non-polar kind of molecule, right? So we are basically making use of the nature or property of my compound to separate it. Mobile phase will be polar, stationary phase will be non-polar, okay? Or vice versa. If my mobile phase is non-polar, like hexane, then my stationary phase will be polar, which will be silica. Okay, so it will always be vice versa. Mobile phase and stationary phase will be vice versa, so that we can separate our compounds of interest. Okay, there will be partition between my mobile phase and stationary phase. So that was little bit basics, how we do the separation or what is the objective. Okay, how the compounds are getting separated based on their polarity. If any compound is coming out first, eluting out first, which means it is polar in nature. Okay, any compound which is eluting last, it is non-polar in nature. If we are using reverse phase chromatography, and 90 to 95% of my work is with reverse phase only. Okay, so before we move to the applications, little bit of instrumentation. The binary systems are there, okay, with very simple configuration with a choice of detector. Then the another binary systems are there which have a capability to go up to 20 ml flow rate. Like if you want to do some purification, like sir mentioned about the phytochemical applications or phytochemistry kind of compounds. So you want to purify the compounds. So you can inject more and you can purify the fraction, collect the fraction and then evaporate it and collect the pure compound. So for that, these extended flow kind of instruments are available. Similarly, auto purification kind of systems. These are mainly used in, again, the industries, like TCG is here, or some other industries like Jubilant is there, Syngene is there, where they want to purify in huge quantities, in grams, if they want to purify. So they use these purification technologies. This can be mass directed or this can be UV directed. Okay. Coming on to the detector part now. Okay. So when we say detector, there are different kind of detectors available. Okay. In the HPLC domain, the role of a detector is basically to sense the presence of any constituent or compound and to give the signal to the electronics. That is the role of the detector. Okay, the output what we see as a peak or a chromatogram. There are different kind of detectors available. The most important role or the most important thing when we are choosing a detector is to look at all these things. The most important is chemical property of my sample. Okay, what is my compound? The nature of that compound. Is it chromophoric in nature? All of you have learned chromophores? Chromophore, non-chromophore पढ़ा है? Yes, no? याद है? भूल गए? Woodward-Fieser rule याद है? Anyone? Lambda max कैसे निकालते हैं? Third year chemistry? No? निकाला है lambda max? Wavelength based on the structure? And Woodward-Fieser rule. So based on that we can calculate what is the wavelength at which this molecule will absorb, there should be a functional group, okay, a double bond, a functional group, a methyl group, a ring has to be there. So based on that, the molecule has its nature. So chemical property of the molecule, okay. Then mobile phase constants. So what are the mobile phase which can be used? So the solubility of your compound is very, very important, okay. So the basic thing, usually what we start our research work is that we take the sample or compound of interest, we solubilize it, right? We check where is the solubility, in what, basically mobile phase or solvent, and based on that, then we choose our technique. Another part is whether I need isocratic or gradient, okay. Isocratic is if it is a pure compound, okay. Like say for example, all of you know about paracetamol, yes, acetaminophen. So if it is a pure compound, they say 500 mg, okay. So there is a assay test for that, right? So purity check is made and that percent is calculated. So if it is a single compound, usually we use isocratic method, okay. If someone comes and tells you, okay, in this paracetamol, you identify what are the impurities which are present, okay. I am not interested now in the main peak, I am interested in the impurities, okay. So if I run isocratic, it will take long time and all the impurities may not elute out, because the nature of impurities will be different. Some will be polar, some will be non-polar. So in that case, what I do, I use a gradient, okay. From polar to non-polar, I run a gradient and I elute out all the impurities. So if it is a mixture of compounds, like your natural product profiling when you do, so mixture of compounds are there, so we use gradient, okay. Then limit of detection, how much I want to detect, identification of peaks. So all these factors will play a role when I want to select the detection technique, okay. And there are different detectors available. Again, 90 to 95% of work is being done using these two detectors, okay. UV visible detector and PDA detector, photodiode array, okay. If the compound is chromophoric in nature, we will be using UV visible or PDA, okay. If the compound is non-chromophoric in nature, which means it doesn't give response to UV visible light, then we need to use other detectors, like our fluorescence is there, conductivity is there, light scattering is there, or in case of sugars and carbohydrate analysis, we use a refractive index detector, right? रिफ्रैक्टोमीटर यूज किया सबने, होता है यूज, अभी नहीं होता, एबेज रिफ्रैक्टोमीटर, नो, इन ग्रेजुएशन फॉर शुगर एनालिसिस एस्टिमेशन ऑफ R and L, yes, R and S, you might have used it, theory में पढ़ा होगा. So on that similar concept, this, pardon? Okay. So on that similar concept, this refractive index detector is built, okay, RI detector. Similarly, light scattering detector, ELST, okay. So when I say UV visible detector, UV visible basically is for chromophoric compounds and where we know the wavelength. Now if I want to analyze paracetamol or acetaminophen, okay, or any natural product compounds, so usually I know the wavelength, I know it will absorb at 254 nanometers. So I put the wavelength, I can use UV detector, okay. But when I am doing research work, I am working with unknown molecules. So at that time, I don't know the wavelength. So in that case, UV visible will work. But if I have a photodiode array, PDA, okay. So in that case, PDA will be much more useful. So that is the reason you will find that in case of R&Ds, be it pharmaceutical or other chemical industries, in the R&D, usually they use PDA, okay, photodiode array, in which you can scan the entire wavelength. You need not fix any wavelength, you scan entire range. Whereas in case of quality control, QC, we find lot of UV visible detectors because they are not doing any R&D. They are only following the procedures, fixed wavelength, and they are doing the analysis. So in case of QC, it is UV. In case of R&D, it is PDA. Okay? स्पेक्ट्रोफोटोमीटर यूज करते हैं आप लोग? यस? पहले आप क्या करते हैं? वेवलेंथ निकालते हैं, मैक्सिमम एब्जॉर्बेंस, राइट? एंड देन यू फिक्स दैट वेवलेंथ एंड डू योर एंटायर स्टडी. This is what you do. Similarly, if I am doing R&D, if I don't know the wavelength, unknown compounds, I will use PDA. It will scan the entire wavelength range from 200 nanometers to 800. UV visible range all. And once I know the wavelength, based on the response, then I fix the wavelength, then I use UV detector. Okay? Then refractive index we discussed, especially for sugar carbohydrate kind of applications. Then fluorescence for some of the very specific molecules like amino acids, or some pesticides are there like glyphosates, which don't give any response to UV visible. So we have to use fluorescence. Then light scattering is there, and finally mass spec detector. So based on what my purpose is, I basically choose what detector to use. Okay? This we can skip. This is UV visible, and this is PDA. Okay? The role of PDA, photodiode array, will be basically here. When I am doing my method development, I need to check the purity of my peak, whether my compound is pure or impure. I need a PDA detector. When I have a pure compound, I can do library matching. I can generate my own library. For that, I can use a PDA detector. PDA stands for photodiode array detector. Okay, which basically scans the entire wavelength range. This is how the data will look like. This is from a natural product extract. If you look here, there are different peaks. Here we are seeing on the x-axis is my time, run time, and on my y-axis is my response or absorbance. For each of the peak, here I am getting a PDA spectrum. Can you see different wavelengths here? This is 245, 239, 240, 240. So some 3-4 compounds are there which are of the same wavelength. Okay, but because their retention times are different, so we can easily identify those compounds based on their retention times. Even if their wavelength is same, but their retention times are different, we can identify those compounds without any difficulty. And we can generate our library or database and confirm that compound from the library. So this is peak identification. Next is peak purity. We can identify whether my peak is pure or impure. In case of mainly the industries, they need this peak purity calculations to ascertain that there is only single compound here. It doesn't contain mixture of compounds. Okay? Then that was all about traditional LCs. Okay? So any questions? All of you are aware? Yes? No? Answer ही नहीं आ रहा है, मुझे लगा सब सो गए. Okay. So when we talk of traditional liquid chromatography, okay, and now moving on to the advancement in the liquid chromatography. So just moving like before moving, quick questions, okay, फटाफट आंसर देना, okay, otherwise I will keep on asking questions. Okay, so just come out of your routine studies, laboratory work, okay, just think about around you in the last 5 OTT communication. Yes. How many of you go to cyber cafe? No? Why? This generation must have not heard of cyber cafes. Yes, I was about to ask that. We used to go in cyber cafes, there used to be a queue. For half an hour, we used to pay 20 rupees. Yeah? Nowadays, just open the mobile phone and everything is connected. Right? Is there a power cut here? Sometimes? Yes? In the night, if power cut happens, what you do? The first thing. Pardon? What is the first thing what you do? Yes? Yes, you are right. So, stop using or start searching for the phone. Because now my brain knows that there is a torch in my phone. Yes? Now my brain no more searches for a candle or for a torch at home. Because it is being tuned that way. Same way for calculator. What is the time? From where you saw? Yes? So we don't use watches nowadays, most of us. आप लोग यूज करते हो स्टूडेंट्स हो तो क्लास कब खत्म हो रही है, राइट? So this is how technology has basically taken over our lives. We may not realize it, yes, but knowingly or unknowingly, it has all taken over our lives. Lot of things are good, but lot of things, there are always some challenges and apprehensions. Okay, now coming back, all of you are awake now? Okay, great. So coming back now, we discussed about traditional techniques. Moving on quickly to the advancements, right? So when we talk of advancements in our life, okay, things around us, lifestyle, surroundings, everything. So similarly, the technologies what we have been using or have been heard for so many years, they are also changing a lot. So routine LCs, okay, to a newer technology known as UPLCs, okay, ultra performance liquid chromatography. It is not something new, it has been there for the last 20 years. This year in May itself we celebrated the 20th anniversary of UPLC. Then a newer technology, because not everyone was very comfortable with UPLC. So a newer technique or a hybrid kind of technology known as UHPLC, ultra high performance liquid chromatography, came into picture. So what is the basic purpose? Resolution has to increase, that is separation. The main objective of coming out with these technologies is time. Overall analysis time is reduced. So when I want more high throughput, more output, I need to go with faster technologies. Okay. So when we look at the different technologies, the dependable LCs which are always there, then there are some new LCs coming up which are known as flexible LCs. So you can use it as a LC or a UHPLC. And then there are UPLCs which are performance LCs. Okay. So, sorry. When we say UPLC, what exactly it is? This is little bit of theory. It is basically a new category of separation science based on the same theory and principles of HPLC. So our HPLC basics and HPLC principles remain same. Only changes have happened in the particle size, that is column, system and detectors in the activity UPLC domain. Okay. So for faster high throughput. If you look at the HETP, height equivalent of theoretical plate on the y-axis, and on x-axis is linear velocity. In HPLC, we use different particle size columns, okay, 5 micron, 10 micron, like that. Whereas in case of UPLC, we use 1.7 micron particle size column, smaller than our hairs. That is the particle size of that silica which is being used, okay. So this is just an example how fast things have become. This is a normal HPLC analysis method of 55 minutes. When I do the same sample analysis on UPLC, 14 minutes. So in the same time, I can do 4 samples if I do the analysis on a UPLC. So if I want high throughput, especially from the pharmaceutical QC kind of environment or R&D, I can use this UPLC techniques, okay. So then finally quantification. If I want to quantify my compound, for quantification we need a standard. So I prepare a calibration curve, and the calibration is always done using the area under the peak. It is not done using the height, okay. So area under the peak, different concentrations will give different response, and based on that we plot a calibration curve, and based on that calibration curve, when we inject the unknown sample, it can be identified accordingly, okay. So we can do qualitative work as well as quantitative. Both kind of applications can be done. And we can also do multi-quantitative analysis. Multiple components are there, they can also be quantified, okay. So this is just the scoring report and the software aspects. This we can skip on. So quickly we will move on to the next session, that is with respect to the applications and with respect to the mass spectrometry. Any questions so far? We are good. I tried to make it very basic so that all of you understand it and we are all in tune. Anyone? Any quick questions? Yes. Purity with respect to? Purity is similar. If purity is not similar, then the purpose is defeated. So the purity is similar. Only the difference is in the efficiency, the fast balance. Purity is similar. Even the profiling is similar. So what usually they do in the industry is that they do comparative analysis first. In HPLC they run the samples in triplicate. In UPLC also they run the sample in triplicate. They compare the results. And based on that, then they move the methods to UPLC. Any more questions? Any questions? No? Okay. So coming on to another interesting aspect, HPLC, all of us probably are more familiar and will be using it during our either master's work or PhD work or after master's when we go to the industry, okay. But mass spectrometry is still a very, very niche area and very specialized field, okay. If you learn mass spectrometry, then you will be probably what we are seeing in the industry, you will be valued more. Because out of 10 people, okay, 10 people who are doing HPLC, out of that only one person is doing mass spec. Probably that is the ratio as of now, okay, in the industry what we see. But the applications in industry are increasing day by day. So little bit again basics about mass spectrometry. First of all, we need to understand why we need mass spectrometry, okay. If I want to identify a unknown compound, okay, a unknown peak is there, I want to identify it, so I will be using mass spectrometry for that. It gives me the molecular weight of a chemical or a biochemical species. So basic chemistry, pharmaceutical chemistry, biochemistry, biotechnology, microbiology, pharmaceutics, proteins, metabolites, everything have mass, so it can be identified. Then unknown compounds, structural elucidation. You might have studied about NMR. Yes? Similarly, mass spectrometry is a complementary technique to identify the structure of that compound. Okay? That structure of the compound can be identified. Then it gives unambiguous identification. It is highly specific, has a wide dynamic range. When we say dynamic range, which means trace level quantitation to purity level, everything can be identified and quantified. That is dynamic range. So historically, mass spectrometers were bigger than humans. Okay, probably this was 30-40 years back, where a lot of manual things used to be there, lot of tuning. For 2-3 days the machine had to be tuned. It was so complicated. Okay, it was large, lot of manual calibration, manual things have to be done. Now the mass spectrometers are becoming much more simple, more sophisticated, more automated. Okay, and probably going forward, a lot of work has been put in. I will discuss in detail also, in which we are having smaller and smaller mass specs for routine applications. So little bit of again basics. The most important thing in any mass spectrometer is how do we identify the mass. In any mass spectrometer, it identifies the monoisotopic mass, which means the isotopes are also identified. Okay. So the mass of an ion, it is calculated using the exact mass of the most abundant ion, whatever is there. We don't calculate the nominal mass. The mass spec gives you the monoisotopic mass. Okay, that thing always you need to remember. Then what are the different constituents or components of a mass spectrometer? We will have a sample introduction technique like a LC or a GC. Then we will have ionization source where the compound or molecule of interest gets ionized. We basically apply a high voltage, 3 to 4, 5 kilovolt is applied, so the molecule gets protonated, M plus H, or molecule gets deprotonated, M minus H. Then we have different mass analyzers followed by detectors. And finally the data system. So these are the different components of a mass spectrometer or a LC-MS-MS system. So electrospray probe tip. So what happens is, when I am connecting this to a liquid chromatography, in liquid chromatography we are using mobile phase, methanol, water, acetonitrile. So all that is passing through this tip and coming out. Okay. At the tip of this capillary, we are applying a very high voltage of 3 to 5 kilovolts here at the tip. And from the sides, we are giving nitrogen gas, and that is also heated. So what will happen, slowly the droplets which are there of our solvent, it start getting evaporated because of the higher temperature, and it forms this plume kind of a thing, a spray or a mist. This is how it looks like. This is the probe, heated wire, and at the tip of this we are applying very high voltage. So solvent starts getting evaporated. Here we can see droplets which are getting formed, and in those droplets our compound also is there, molecule of interest, either a positively charged or negatively charged. And as they move forward, there is a temperature, high temperature, 400, 500 degrees, so it starts getting evaporated. So what will happen? Droplets starts becoming smaller and smaller till the time the solvent is completely evaporated and only my compound of interest remains. That's it. And that is going to the mass spectrometer. So basically we are doing ionization followed by evaporation of the solvent. After evaporation, only my compound of interest is remaining. which in a form of ion, M plus H or M minus H, is going inside the mass spectrometer. Okay. So what are the routine samples? What applications we can do in mass spec? So peptide, proteins, small molecules, drugs, metabolites, some dye compounds can be done in positive mode. In negative mode, we can do some protein metabolites and some saccharide, polysaccharide, sugar kind of moieties we can do. Okay. So how basically this works? If you look here, this is a lidocaine molecule. Okay. If you look here, it has got this nitro group, okay, amide, and here also it has got a secondary amide. If we do it in a positive ion mode, nitrogen, all of us know, can take up a lone pair of electrons. Okay. So M plus H. So when we protonate this, nitrogen can take up another hydrogen. It can form a positive charge. So we will be getting this as M plus H. If we analyze the sample in negative ion mode, this is ibuprofen. Ibuprofen has a carboxy acid group, COOH. Okay. All of us know that OH bond is weak, right? That hydroxy bond, that H can be removed very easily. So we can easily deprotonate this. So what will happen? O minus. So M minus H. So whatever your molecular weight is there, minus 1 will be the peak what you will be seeing in a mass spec. So for a successful mass spectrometer, the most important condition is that the compound should have a functional group. Okay, it can be a carboxy, hydroxy like COOH, OH, or it can be a amide group, secondary amine, anything. It has to be there for a successful LC-MS analysis. Okay. So coming on to the LC-MS aspect now. Okay. Some of the applications when we discuss. So this is the smallest mass spec available by the name QDA mass detector. Okay. Which is basically used for routine applications like your purification or routine mass identification. It can be used. This is all adjustment free. This is based on the detector concept. Everything is automated. There is a calibrant inside this. It can automatically calibrate itself and run it. So what are the different applications? Here I will like to spend some more time. If we look at different applications like synthetic chemistry, small molecule, okay, or impurity profiling, any natural product profiling, I want to check different sources of any particular natural product. Okay, fingerprinting, what we call. So all that can be very easily done using this QDA mass detector. Another application is in the purification. If I want to purify a compound and collect that compound based on its molecular weight or mass of that compound, that can also be done using this QDA mass detector. Then synthetic compound, the basic application, like if you are doing some R&D experiment, okay, is that I want to confirm the mass of the compound. I can confirm it using this mass detector. Very simple, because in HPLC we need a standard for confirmation, but in mass, based on the mass of the compound, we can easily confirm the standard. Here we can see this 321 mass and this 309 mass, which means these are two different compounds which are present here. Then along with my HPLC, I can also have this mass data. Okay, this is the beauty of this QDA. It can be coupled, and in the same window, same chromatogram, you get PDA data, you get mass data, and on each top of every peak, can you see the molecular weights? 152, 195, it can label them accordingly. So in the same sample like purification, I can have these compounds separated. Then I can also collect these compounds for fractionation. If it is a natural product extract or a pharmaceutical, like you are doing reaction monitoring, a microbial kind of biotechnology kind of work where you want to identify different compounds, you can easily identify them. Then in the method development, when you are doing method development studies, we do different experiments, right? We use different buffers, pH 3, pH 5, pH 7, like that. So when we change the buffers, what will happen? The peaks also change, elution pattern will change. So when the elution pattern changes, every time we need not inject the standard. If I have the mass detector with me, based on the mass, I can confirm what is the compound, what is my molecular weight. So it is very useful when you are working in the method development domain as well. Then assigning the right peaks. In this case, here we can see 300, 316 mass, 286 mass. And this is the PDF spectrum, this is the mass data, 286, 300, 318. So different masses, they can be identified using this mass detector. Then coming on to another interesting mass detector, this is known as radian. Okay, radian ASAP. Anyone? What is the meaning of ASAP? As soon as possible. Okay, so something similar. In this radian ASAP, there is no need of any sample preparation. Okay, whereas in case of other mass spec detectors, you have to first solubilize the sample, filter it properly, and then inject it. Whereas in this case, radian, it is our simple melting point analysis. करते हैं आप? Melting point, yeah, melting point. नहीं करते? No one? Graduation में क्या होगा? Yes, on to one. Science cube होती है, right? Triangular. Then we put paraffin in that and determine the melting point. So that melting point capillary is there. The same melting point capillary is used for taking the sample and doing the analysis. So how fast this is? This is a novel mass spec, again a very small mass detector, specifically designed for rapid, easy and low cost sample analysis. No sample preparation, zero. Just take the sample, dip it into melting point capillary and put it into the mass spec. That's it. So no sample preparation, no training. So non-MS experts, they can also do routine analysis in this. So clean the capillary, this is this melting point capillary, load it with the sample and put this into the mass spec. That's it. It will give you the compound identification. This is a very good example here of reaction monitoring of 4-HPA and atenolol. Okay, atenolol is our final product. 4-HPA is my starting compound. Okay, so initially what is happening at the start of my reaction? Starting compound is more, this finished product is less in 5 minutes. After 20 minutes, what is happening? Can you see the difference? My final product is increasing. After 50 minutes, my final product has increased a lot and starting material is remaining very less, which means my reaction is almost complete. So this can be a very good tool for reaction monitoring, for routine mass analysis. Then in chemical industries, they can use it for testing of raw materials, formulations. So lot of diverse applications are there. This I can skip. Then coming on to that was about routine MS analysis. Little bit more complexity now. Coming on to the triple or tandem quadruple application. Okay. Most of you might have heard about nitrosamine. सुना है किसी ने? Anyone? Quickly, can you raise your hand? Nitrosamine, सुना है? Yes, no? No? Yes? monitored at a trace level, not at a purity level which HPLC can do. So we need a very high sensitive LC-MS-MS. So this is where these tandem quadrupoles will come into picture. Depending on their applications and sensitivity, these different technologies are there, like the routine one TQS, Chronos and Micro, or for very high-end sensitivity, we have this excess and absolute. Okay, I will not go into the product detail, but we can do this multi-mode ionization, in which this is again useful for method development, electrospray positive, and APCI, atmospheric pressure chemical ionization, can be done. This is how the chromatogram will look like. In PDA, we can see a small peak, and in mass spec, we can quantify these compounds at a trace level as well, at PPB and PPT levels. Then coming on to the last piece, that is high resolution mass spectrometer. Okay, if you are doing novel research, okay, very high-end research in the area of, you can do natural products, you can say advanced chemistry or metabolomics kind of work, then these high resolution systems are useful basically to identify the compound very, very accurately. Okay, so here we can see this is resolution and this is also resolution. Which one is better? Eye test? Which one? Yes. So this is what high resolution mass spectrometry will do for you. It will identify the compound accurately and precisely. That's what the message here is. Whereas the LCMS or LCMSMS, it will only quantify the compound, which means it is for known compounds. If you want to identify unknown, we can either use a QDA or a HRMS kind of a thing. Similarly, another example, all these three compounds, they have same molecular weight. Okay? But their formulas are different. So if I identify these in a routine MS system, I will not be able to differentiate between them. Okay? But when I have a high-end HRMS system which identifies mass up to 4 decimal places. So what will happen? I can easily differentiate between these compounds. So this is where the applications or identification in case of natural products, in case of advanced chemistry research, in case of metabolite identification, all these applications are being used. Then another concept is of mass accuracy. So resolution and accuracy, both of them, they go hand in hand. The mass accuracy is less than 1 ppm here. How accurate it is identifying the mass. This is HRMS, high resolution technology. Okay, so smart MS, nowadays everything is smart, right, like our smartphones. So automated setup and push button start. This is how it will look like. Very small footprint. This is a UPLC and this is a small MS detector used for diverse applications. So this I can skip. So I'm just sharing my mail ID and phone number. If you have any questions, queries, or need to connect with any of us, Debashish routinely visits here. He is our local salesperson here. So any questions are there, you can reach out to your students as well. So we are now open for questions. I hope it was useful to you. I tried to make it very simple as well as a little bit interesting. Any questions, clarifications, doubts?
I would like to ask you one question. You have talked about identification of source of different compounds from natural product. So how do we get to do that through MS?
Basically, what is being done is fingerprinting is being made. southern part of India. Okay, that also I am collecting same part, either it can be bark, stem, flower, leaves, anything. I do the same profiling, same process. I identify the biomarkers. What we do is there are different softwares available in which they have a definitive image, and based on that image it will rule out what are the similar constituents. Okay, because we are not interested in similar. It will create an image in which only the different constituents which are there. Some compounds may be present in higher concentration, some may be present in lower, or some may be absent. So it creates that profile, and based on that profile, which are known as biomarkers. So we can then identify that in this source, in this species, these are present. So if we do unknown samples, okay, extract, there are claims available that this is from this source. A lot of studying has been done in this ginseng, okay, because Chinese ginseng is the one which is the most effective, but then Korean ginseng and all other ginsengs are there. So there are some biomarkers which are identified, and based on that, then it is confirmed. So we can do that profile.
So basically it is like you need to have a standard based on the identification that has been done.
No standard, no standard, no standard. Two different set of experiments, two different sources. One say from northeast, one from south, one from north, like that. And we do the fingerprint profiling of all of them, like what we do in respect to the genetics or DNA fingerprinting. So similar kind of work. And on QDA, we can only identify to a certain extent, but we need a high HRMS kind of system if you really need to have a control.
Okay, then say like in context of nutraceuticals or substances where you have mixture of different compounds,
Yes.
aiming from different natural products, then in that case how can you go about it?
In that case, then there are markers identified and confirmed for each natural product. Say for example, like there is tulsi, okay, or like neem is there, so azadirachtin is there. Along with that azadirachtin will be there everywhere, but along with that there are other constituents. If it is from Melia azadirachta, there is also similar species. So based on that, it is identified.
How do we go about it? Means, say for example, I send it to you for analysis, and then after I also need to request you that get me the natural source of it, and then you get back to me likewise. Or how does that happen? The process. Means usually what happens whenever we send a sample for LCMS analysis or your inside LCMS analysis, they give us a library report. After completion of the entire process,
Right,
they will
right.
give us a library report
Right.
along with the structure and all.
Right.
Now I get to know the compound.
Compound,
I get to know the compound.
Right.
Now my constraint is that my sample that I had sent you was actually a mixture of different natural products.
Got it. Right.
It is not only one.
Right.
Maybe it is a microbial metabolite. It can be a metabolomic study. In my research, it is a metabolite.
Okay.
It will be metabolite. So I need to know whether it is a compound from the plant that has been used or the microbes that has worked on the plant.
There are actually libraries available for this kind of work. There is this traditional Chinese medicine library available for natural products. There is public domain like spider databases available for confirmation of all these
So
compounds.
individually against the compounds, I need to compare it with the libraries available.
Any other questions? Good, then I think we can over to you. Thank you so much, everyone.
Thank you very much, sir, for your informative discussion. Even though, say, like we would have like to have questions from the students. You are being master students and going to start or have been doing your dissertation. We expected more questions and more queries from your side. But anyways, he has already left his email ID, and Devashish sir is also here. In the due course of time, I believe you people will be discussing more with your supervisors regarding your dissertation work. and then you will get to know more about the processes that you need to carry out in your researches. So in days forward, if you have any queries and if you need to do any such analysis and if you have some problem in analysis of those compounds, then maybe this, they can help you in this regard. So once again, I would like to extend our gratitude and heartfelt thanks to both the officials from Walters, and we hope to have more such events and more such, maybe a workshop next time, wherein we can have some portable machinery so that students can go through those, and if we practically do the things in a workshop mode so that they can analyze the peaks. The problem, what happens in theory is like we have been delivering these lectures, again we do this. When they get to see it practically, then it becomes much easier and it becomes much interesting for them to say like dive deeper into it, and maybe then I believe there will be more questions from students, and once they practically understand everything, I believe they might have not understand some of the things that this happens because there are technicalities which they might have not come across as of now. So maybe we will try to have a workshop later on with discussion with Walters, and maybe we can have say proper discussion and analysis, the entire process of analysis. So once again, thank you all very much for your patient hearing. We would like to continue this effort. Suman sir is the coordinator. He has, you should all clap for him that he has coordinated. He has brought sir from Delhi. And we also have some participants from Arjo Vidyapit. I believe the people have come from Arjo Vidyapit, and you are welcome. We are looking forward to hosting more such events wherein we can have scholars and we can have industrial experts from different industries across India and abroad. So thank you very much.